Great. Okay, so we have the downloadable PDFs. Same thing that's on D2L in the thing where it's information like review information. Okay, I'm recording. Review information for the final, what to study for the final in the unit materials. So I have the two of those things here. There's other ones in there, and then I've got some links back to the old boards if you want to go to those, and the link to the practice board, so everyone's going to get a practice board, so you can of course click on that and get over there if you want to. And then I've expanded the PDF of the like kind of review questiony things, topics I suppose, and then the slides as well, and we'll look at some of those. I'm going to focus on this review mostly for the beginning stuff because I think most people do generally quite well on animals 2 in particular, which we just did, right? So I'm not going to re-go through that. And we really won't focus on animals 2 at all, even though of course you will need to, know that information. And then animals 1, that's been really quite recently as well. So I'm going to mostly focus on reminding us about some evolution ideas, and then the protists and prokaryotes, just some of that stuff. And then we'll kind of like incorporate sorting into tables, certain things, and to see how we can compare the groups, that kind of stuff. So I've also embedded some videos in the slides, or put links to, or some videos and stuff like that. So you can look through those later as well if you need any of those animations to give you an idea of some of the concepts. Not going to go over natural selection really, because I think you guys pretty much know that at this point. We'll give you a reminder of some of the microevolution stuff, and then a little bit of speciation stuff and what you need to know for that. And then, like I said, I do want to largely focus on that prokaryotes and protists, Unit 2, which most people struggle most on Unit 2. And then we'll remind ourselves of the life cycles of fungi, plants, and animals, and then the phylogenies, because you will have phylogenies. There's not a whole lot about, there's not tons and tons of stuff about plant physiology or anatomy and physiology, like the plant parts. There's some of it, but it's not like super detailed. You're not talking about minerals. There's nothing about hormones or anything along those lines. It's pretty basic and straightforward, the different types of growth and the different layers of the plant, the different organs and what they do, that kind of thing. But of course, in the review, you do have all the information, or here's what you need to know for plants, basically. So same thing for all the groups. Okay. Are there any topics that anybody was hoping to review that I should just make sure we don't run out of time for?
Can we review plants?
Some plant stuff?
Yeah.
Anything specific?
Probably the anatomy.
The anatomy, physiology, okay. And we can look at the questions here, because I don't have too many plant slides in here, but there are some pictures in the little review thing. So yeah, we can talk about those, and monocot, eudicot, the plant groups for sure, and the features of those different groups, and their ecological kind of, or their symbiotic relationships, that kind of thing. Okay, we'll make sure we do that. Okay. Obviously, you'll need to read phylogenies and interpret them. So not just filling them in, but interpreting. I feel like we're pretty good at that now, so I'm not going to really go through that. These slides are separated into sort of topics. Let's see. Sure, go ahead.
Sorry, on your so in the final review sort of stuff that you have going on, it explains which phylogenies and how much detail we need to know about them, because on the review itself, it says be able to draw the phylogenies for the 3 domains of life, and Archaeplastida and Uniconta. So is it going to be like we're going to go over the more specific ones, like deuterostomia and non-deuterostomia? Like we're going to get into that more specific stuff, like we did with our past units.
Yes, you will want to be able to basically all the phylogenies for all the different groups of what we've done before to the level of detail that we've done. Nothing more detailed than what we've branched it out to be, but yeah, like all the kinds of things.
Yeah, so like the synapomorphies, the example species.
Yeah, yeah. Okay, space should be
Oh, and
upgraded now.
this is a kind of unrelated question, but we're gonna have like short answer questions, kind of similar to what we normally.
No writing. I think there's no writing. There's a few places where you have to enter a letter, like A, B, C, D, E, out of a choice bank. But yeah, I don't have any kind of short essays.
Okay.
Yeah, for I don't have that much time to grade it,
that's fair.
Yeah, and then you guys, have done a good job showing me your writing already in this semester. So.
Yeah. So, do you know about how long the test is? Like, how many questions?
Yeah, so it has quite a lot of questions, is what you're going to notice when you go on. But I do want to point out that many of them have it's really just filling out a phylogeny, but it's like 15 questions to fill out the phylogeny, right? Because sometimes it's like each answer is a separate question. It has to be like that sometimes. So it's not quite as many as it's going to look like when you see the number. I think it's like 120, which looks like a crazy big number. But like I said, if you were just filling it out on a piece of paper, you'd just be writing in each answer, right? Which would just be like one question. So it's not quite as many. I would say probably real question wise, probably like 90 maybe. Something like that. And then there's lots of extra points, 15% extra points like we usually do. Okay. Well, I think that we should maybe skip evolution for the moment. Unless there's something people specifically wanted to do. Natural selection we're okay with. Let's see what else. Phylogenies. Oh, I guess I will say this. All right. So let's just remind ourselves about, I'm on the slide, sources of genetic variation. Okay. This is on the slides you can follow me, of course, the second row. Okay. So this is, just to, remind you about the sources of variation, because you have to have variation, of course, before you have the ability to have selection happen and a shift in populations, right? Because if everything's clonal, while that may be advantageous in a stable environment, it's not going to allow for natural selection to occur, or even any evolution to occur, even if it's by there is no, even if there was like a catastrophe event, there would be like a genetic drift bottleneck. It can't do it. There will be no shift because everybody is exactly the same. So the genetics will stay the same. But in populations where you have variation, of course, then there's options for the shifting of the population towards different characteristics. So just to remind you, mutations, which is usually what people think about, and that is the initial source that you get the variation in the actual DNA. But remember, we can also, and this goes along with mutations, but often what you see is altering this gene number and position, where you're going to have the gene get copied by accident during DNA replication. And then now there's two copies of the gene, and one can diverge or change the sequences through mutations in one way. And the other one can, maybe just stay the same, even though they both accumulate mutations. It's like if you have a scenario where you get mutations in one but not the other, maybe the organism doesn't die from those mutations because it has a good, a working copy of the gene still. So that you see that often where you get this copying and then divergence, like all those Hox genes we've talked about now. There's many copies of the Hox genes to do the body plan in animals. And each of those diverge slightly from each other, and not just within the population or within the lineage, but it's actually between all animals. You can look and see these copying events throughout the evolutionary history of the animal. Speciation and divergence. Sexual reproduction, as you know, really important variation source. So it's like the combining of the egg and sperm, which are, both have different genetics. So that is a combination. That's random fertilization. We've also got the crossing over in meiosis and the independent assortment of chromosomes in making the gametes. So both of those in making the gametes and then the gametes coming together and mixing together are sexual reproduction. Gene flow, remember this is only bringing in genetic variation if it's gene flow into the population. So you're not going to be getting new variation if you're gene flowing out of the population. It's only when you get new individuals in with new versions of the alleles. And that's going to bring new variation of the population. And then this lateral or horizontal gene transfer, I don't have massive amounts of this, but I did want to remind you, if we go to the next slide, looking at this horizontal gene transfer, just the three types, the transformation, transduction, and conjugation. So I'm not going to ask you in massive detail about all the steps, but it would be good to have some idea of the difference, right? So transformation is when the bacterium picks up DNA from the environment, like fragmented DNA from the environment, and incorporates it into its own genome, which brings new alleles in and do new combinations into its genome. And this is different than reproduction, as we know, because reproduction is when you make an offspring. This is not making offspring. This is like changing the individual bacterium as it exists. That's horizontal gene transfer. And then just transduction. Remember the DNA is, or the genetic material, I guess it would have to be DNA, actually. The DNA from one bacterium to another, it's accidentally transferred by a virus because the virus has broken up all the DNA inside of the host cell. So some of that DNA gets picked up by accident when the virus is being made and then gets transferred. And when it infects the other bacterium, then it puts that DNA inside that bacteria cell. If that bacteria cell can recombine it into its genome, then it'll have potentially new combinations of genes in its genome now. So transduction is with the viruses. And then the conjugation, co means together. So this is the direct transfer of genetic information from one bacterium to another through the plasmids typically. Sometimes it can be parts of the genome, but we talked mostly about transferring the plasmid, which is a small DNA circular DNA copies in the cell that have just like one, maybe two genes on them. And so the bacterium that can make the pilus to deliver the genetic information, it will make a copy of the plasmid, transfer that copy to the recipient bacterium, and then it's really one strand. Both of them make the new strand, and then they both have a full plasmid in both of their cells. And then they decouple. That conjugation bridge goes away. So that's the direct transfer. So I just want to remind you about that. And then moving to the, you want to know the 4 causes of microevolution. With this, mostly I do something like, here's a scenario. What kind of microevolution, mechanisms are operating in this situation? So you're thinking here, is it a natural selection situation where the most successfully surviving and then reproducing organisms, but generally natural selection, we're thinking about the survival aspect causing the shift in the population because only certain individuals are surviving more successfully than others. Genetic drift, and remember, genetic drift and gene flow, there's some confusion about that sometimes. So genetic drift, remember, it was named because when you look, and you saw this in your sickle cell symbio, when you looked at a population, if it's small, so this is the genetic drift, when we're talking about small populations, the genetic drift affects it more because all it is this kind of like random events that change the allele frequencies. And the name drift came because they watched these populations and then they would see sometimes this is the allele frequency. The allele frequency just drifting down from, maybe 50% here to like 1% there. And there was no reason for it. There was no natural selection going on or anything like that. It was just a small population. And all of a sudden they just saw the frequency of a certain phenotype and also ultimately a certain allele just like drifting to being very common or just drifting to being very not common. That's where the name drift comes from. Gene flow is that physical movement where you're actually getting individuals coming in or out. And that, of course, makes sense that it would change the allele frequencies, the frequencies of the, versions of the genes, because if you're leaving, you're taking your genes with you, or coming in, you're bringing new genes or new copies of the gene into the population. With genetic drift, remember there's also like the two, well, there's maybe three-ish different kinds of things I think of with that. One is that this is all like a random chance kind of thing. How do I want to say this? So basically, it's like, what do I call it? When you flip a coin, you're supposed to get 50-50, right? Five heads, 5 tails for 10 flips, but you don't always get that exact distribution, right? There's just variation in that exact perfect scenario, especially when it's only a small number of flips. The same thing here with this genetic drift. If you have a small population, yes, you should get 25% of the offspring being white flowers and 75 being purple flowers, whatever, something like that, with math. But does that exactly happen? No, because it's not perfectly, everything's not like perfectly random and perfectly 50-50 kind of thing. So we would say something like, it's not perfect, though. That's not the right words. Random chance, I guess. I don't know how to say this. I'm sorry. I forget what they call it. I'll look it up later. Random chance in genetics outcomes, where it's just like not perfect in the mating. Then the other two that you do know are called the bottleneck effect. Or the bottleneck effect and founder effect. So these are two types of genetic drift. The bottleneck effect is when you have a population that there's a big death event and only some make it through. That's what the bottleneck is. Like if you think of a bottle and you have stuff inside of it, only some of it can make it through the bottleneck, right? Or like traffic, your traffic bottlenecks down to one lane. It's only few cars make it through at a time. And so if you have this catastrophe event, it's bottlenecks, meaning only some individuals survive. It's not the ones that survive that are the fittest in this case. Maybe, maybe not. It's just that some portion of the population survived randomly. Everyone else got killed because of some event. It doesn't have to be like a natural disaster. I mean, it doesn't have to be catastrophic, like the asteroid hitting the Earth. It can be something like, a person stomping on flowers when they were hiking, and the population's small, and so they might just crush certain plants and not other plants, and so then that's the bottleneck. The founder effect is when you have a new population forming from a previous population. So you have, the beetle population with brown and green, it's 50-50, brown beetles and green beetles. Then you got a little group that they're sitting on a log and the log just goes downstream and ends up stopping somewhere else. The beetles get off and then they form a new population there. The beetles that got off the log are not going to be, it's unlikely that it's going to be perfectly 50-50 green and brown that happen to go to this new environment, right? It's like the new population, the founders, like the founding fathers, if you will, the founders of that population are not going to be in the same, or if they're not in the same proportions of genetics as the original population, then your new population will have shifted genetics because of those founders being different there. So you often would find founder effect in you're thinking of, is there a colonization and something new about the colonization, some shift in there? That's probably founder effect. And those are all examples of genetic drift. And then sexual selection is where it's not the survival, it's the getting mates. that you're most successful at. Because in the end, survival doesn't matter if you're not making offspring successfully. And that doesn't matter if those offspring aren't fertile and making their own offspring. So, but the sexual selection, you may not survive very well, but you might get mated with a lot or be able to mate with others a lot and therefore pass on your genes. So that's why sexual selection would cause microevolution. So all we're talking with microevolution is this shift in allele frequencies. And then just to remember the names of the possible results of natural selection. Oh, I have some blanks in here. Okay. So this is like a mouse population that has the colors, fur colors, light to dark in a gradient, basically, is what we're saying. So different selective pressures, and this is natural selection. Different selective pressures are going to shift the population in different ways, of course. So in the first one we have here is directional selection. You can see that's moving the frequency to one direction. So with this kind of thing, I would give you a scenario and ask what kind of selection selection do you expect to happen? Do you remember the name of this middle one where you get the population kind of bifurcates to the two different phenotypes, in this case particularly the extreme light and dark? Or we could say this one, so that's number 2. Or number 3, if you remember that, where it goes to the middle phenotype and it like more and more become the middle phenotype.
Stabilizing?
Which one, 2 or 3?
2.
2. Okay, to stabilize, let's see. Stabilizing means to make steady, right? Like if you're stabilizing yourself when you're, out of balance. So it's making it, not any of these are not, they're all stable. But what I think with stabilizing is it's helping keep it narrower and in one range. That's what I think of it as, because it's keeping it in one spot instead of 2 spots here. So
I'm sorry.
That's
2
okay.
would be disruptive.
Yeah, 2's disruptive because that's okay.
Yeah, I read
2
it
is
wrong.
disrupting. Oh yes, you were looking
yes.
at the solid line being the second.
I'm sorry.
Yeah, dotted line's the new distribution. All right. Yeah. So disruptive meaning it's selecting for two different phenotypes simultaneously, often at the extremes, and then and then stabilizing is that selecting for a middle phenotype that was like and almost like the most common. Whatever was the most common just gets more and more common. So it's that middle one. So I just give you scenarios there. And then I put this little video in here that's just reminding you. It's a little thing about drug resistance. Just reminding you that natural selection, of course, is only acting on existing variants or variants that rise in the population, right? The actual selective pressure, whether it's drug treatment, whether it's predators, weather, climate, flooding, or getting food, getting mates, any of those selective pressures, it's they act on what already exists in the population, right? Those pressures aren't causing new variants to arise. People really have that problem, that mistake in their heads with drugs, or vaccines. I see it a lot where people are, where it's like, oh, well, the vaccine came out, and so then the viruses evolved in order to evade it. But it's really, now that's not how that works, right? It's a viral population with lots of variation. Vaccine comes out and is effective against many of those variants, but not all of them. Or perhaps it's effective against almost all of them, but then a new variant arises. Usually it's already in the population, though. It just becomes more common because it's the variant that makes it. It doesn't get killed by the immune system because the vaccine didn't prime the immune system to recognize it. Because it was a variant already existing in the population, and then that becomes more common and naturally selected for. Same with drugs, right? There are no bacteria that gain features to make them get around the drugs. That's not how it works. The features already exist in the population because there's a lot of variation, and because there's very fast generation time. There's lots of variation created very quickly, so the drugs just kill all the bacteria that are susceptible, but not the ones that aren't. Those guys reproduce, and then you have a population of bacteria now that are not responsive to whatever drug you were giving them. So already in the environment. And do also remember that individuals cannot evolve because they, this is all talking about shifting of genetics. Like an individual is born with the genetics it's born with. So individuals don't evolve, populations can evolve, species can evolve, if we're talking a macroevolution, that kind of thing. So just don't forget that. Okay. I did want to do a Hardy-Weinberg problem because I know sometimes people get nervous about those. There's like a couple questions about, or there's maybe like 4 Hardy-Weinberg related questions. One about the conditions needed for Hardy-Weinberg equilibrium, or conditions that are broken when it's not in equilibrium. So just make sure you review those 5 conditions there. Basically, it's like if you have any mechanism of microevolution happening, you're not going to be in Hardy-Weinberg equilibrium because your population is going to shift. So it's kind of like the opposite of microevolution mechanisms. We have that here, 5 conditions that must be met for Hardy-Weinberg equilibrium. Oops. No mutations happening in the population, or they're very infrequent. No sexual selection, essentially, which is what random mating leads to, that there's no special pairings or more successfully paired or not. No natural selection, everyone's surviving equally and reproducing equally. Extremely large population size, so you reduce the effect of random events in that random coin flipping thing. So that's why you need that large population size and no gene flow, because then you'd be shifting the population. So, and then just remember, if it's in Hardy-Weinberg, you expect those allele frequencies and genotype frequencies to stay the same generation after generation, because there's nothing acting on the population in order to make it shift around, right? All right, and that is our little p squared, 2pq, q squared. So I think I have two problems on the exam to solve p's and q's stuff. P squared, 2pq, q squared equals 1. All this is meaning is that, just to remind you, p is just what we use to mean the frequency of the dominant allele, assuming there's dominant recessive. If there's not, if there's codominance and stuff, then you just use it, you pick which allele it is. But generally we're talking P equals the frequency of the dominant allele, the Q equals the frequency of the recessive allele. And you remember these are allele frequencies, the single letters, because Q is just a single letter, and an allele is just a single letter. Like when you write in a genotype, then little a, that's the allele. So P is an allele frequency and Q is allele frequency. And remember, frequency just means the number of that allele type over the total number of alleles. And then the genotype frequency is the other thing. So that genotype frequency is saying, like, how often do I get the certain combinations? How often do you get 2 P's together? What's the frequency of that? So getting 2 P's together would be the probability, or the frequency, I should say, the frequency of one A times the frequency of getting another A, right? Because those are 2 separate events. And so P times P is P squared. And so that's the frequency, which is the same going to be as the probability, the frequency of big A, big A. And then what's the frequency of getting 2 little a's together? Well, Q is the frequency of one, Q is the frequency of another one, so Q times Q is Q squared, and that's going to be the frequency of your little a, your homozygous recessive. And just to remind you, that's going to be, ooh, that's very thick. One of the ones that you're probably looking for, because Q squared, you can always tell what Q squared is, regardless, like there's only ever one way to get a little a phenotype. So you always know who is a little a individual, because if you're recessive, like we know that you have to have 2 copies of the recessive allele to look recessive. That's what recessive means. So there's only one way to look recessive, having 2 little a's, And thus you know that's q squared. So this is going to be often you're trying to like get yourself to q squared because then you can solve everything. And then what about this? What about the other genotype, the frequency of big A, little a? Well, we have the frequency of getting a big A, P, and the frequency or probability of getting a little a, Q. But then remember we said you can get big A from mom and little a from a dad, or a little a from a mom and a big A from a dad. So it's like two ways to get those together. That's why there's the 2 on the front for the heterozygote. So we just call that 2pq. And that's the frequency of big A, little a. Now, how do you know these are frequencies of genotype frequencies? Because they have 2 letters in them, right? A genotype is AA, or big A, big A, little a, little a, big R, CRCW, whatever. It's 2 entities to make a genotype for a diploid organism. So that's how you remember, oh, P squared is 2P, it's like P times P, that's 2. 2PQ has P and Q, that's 2. Q squared really just means Q times Q, that's 2. Those are genotypes. So when you're working problems, you just pay attention. Are they telling me something about allele frequency or are they telling me about genotype frequency? Let's go down to this problem here. So there's a problem about butterflies near the end of this slides things. It's the easy one to do. So within a population of butterflies, the color brown, which is coded for by big B, is dominant over the color white, coded for by little b allele. So that's the B gene. There's 2 versions of it, one coding for brown, one coding for white. Then they tell us 40% of all the butterflies are white, populations in Hardy-Weinberg equilibrium, and then you want to find, I wouldn't say percentage though. I'm not going to say percentage most likely. I'd just say frequency. But it's just not moving, you just don't move the decimal. So, all right, so what's the percentage of butterflies in the population that are heterozygous, okay? What are we going to ultimately need to find the frequency of? What are they asking us for this A part here? What's the question, like what kind of PQ thing are they asking us ultimately to find to figure out something about heterozygotes?
PQ.
And you know that because heterozygote means a big A, little a, or big B, little b in this case. So 2pq is the frequency of heterozygotes. And that's what we're looking for. Yeah, so that means we're going to need a P and we're going to need a Q to be able to put them in here. What do they give us? They tell us 40% of the butterflies are white. We need to put this in a decimal. So 40%, if you put that in decimal form, what's the frequency of that then?
0.4.
Okay. So 0.4 is the frequency of white butterflies. Okay, so what is a white butterfly? What do we know about a white butterfly? How do you get to be a white butterfly?
Homozygous recessive.
Because it tells us the color white is completely, or the brown is completely dominant. So that means the only way to look like a white butterfly is to have 2 little B's, right? And if we have 2 little B's, what term did are we finding here? What was this is a frequency. So really we're seeing the frequency of little b. What's our symbol for a little letter, a recessive allele, P or Q?
Q.
Q. And this one, we have 2 of them, right? So what did we just find? 0.4, what do they give us? 0.4 is 2 of the Q's, so Q squared. So it's a genotype frequency, right? Even though they give us a phenotype equals genotype. So we know the phenotype is white. We know that means genotype is little b. So genotype frequencies always have 2 letters, which in this case is the Q squared. So they've given us 0.4 is Q squared. So that's not going to help us directly though, right? Because we're trying to figure out 2PQ stuff. So how do you get from Q squared to, I'm pretty sure I know. Yeah, how do you get from Q squared to something useful? 1 second.
Square root it.
Square root it. Yeah, what's that going to give you? What term will square rooting it give you?
Q.
It'll give you Q, which is the frequency of your little b allele. So 0.632 equals Q. Great. And then what else do we need to figure out heterozygote information?
P.
P. And it's easy for P and Q, right? Because P and Q tell you like what portion of the alleles are big B and what portion of the alleles are little b. So if 0.632 is the portion of alleles that are little b, everybody else must have, or all the other alleles in the pool must be big B. So that's just the rest of it, right? So it's instead of 100%, we're just going to call it 1. So, P plus Q equals 1. All that means is like the frequency of the big B allele plus the frequency of the little b allele equals 100%. All the alleles together. So ultimately, we're going to have, we can just subtract that from 1, right? Because it's going to be the rest. So that tells us and honestly, when you're working it, it's not this much explanation, right? When you're working it, you're just like, what do they give me? Ooh, white butterflies. Ooh, that means little b. Oh, that means Q squared. Great. So now I know the frequency of Q squared. Square root it gets me Q. Subtract it from 1 gets me P. And now you've got P and you've got Q, and you can multiply that by 2. And that gives you, the information about the heterozygotes. 0.465. And that will be the frequency of heterozygotes, but I asked percentage here. So in order to turn a percentage, you just, move the decimal over twice. It's because you're multiplying by 100. So we'd expect about 46.5%, or the frequency was 0.465 of the butterflies to be heterozygotes. Okay. The frequency of homozygous dominant individuals. What's the genotype of a homozygous dominant individual?
Big B, big B.
That's what homozygous dominant means, right? Homo meaning same allele twice, and the dominant allele. Yep. So big B is what they're looking for. Recessive allele.
P squared.
P squared. Yeah. So we want P squared. They've given us this 0.4, which we've already figured out is Q squared. Oh, we've already solved for P. I guess that makes it easy in this case. If we were starting from not having P, we'd have to think about it a little differently. Actually, I do want to do that here. Well, you do the same thing in this case, right? Q squared is what they've given you. The mistake that people might make here's what I want to say. The mistake people might make is that they see 40% being white, and then they think, oh, that means 60% are homozygous dominant because they're brown. But how can you be brown? How could you be a brown butterfly genetically?
Big B, and big B, little b.
Yeah. So you have 2 ways to be a brown butterfly. So this is sometimes people like, and you're right that 60% of the butterflies are brown, but you can't do anything with that information because you have 2 genotypes that can get you a brown butterfly. So the key here is always go for the recessive. Just find the recessive. And if we could give you something like 60% of the butterflies are brown. Figure out the rest of it. And you're like, oh no, there's 2 genotypes that give me brown. It's okay. All you have to do is say, well, if 60% are brown, then 40% must be white, the rest of them. And then 40% is white. That means that's homozygous recessive, Q squared, and then you can solve for everything. So that's like kind of the strategies. Generally try to get to Q squared or Q, if they give it, that could be useful as well. because Q squared will allow you to get to Q, which easily allows you to get to P, and then you can do anything. Okay. That's what I wanted to say with the second one. So P squared here, you just, multiply this 0.368, square that. 0.368 squared, 0.135. That'd be the frequency of homozygous dominant individuals. But I want to point out the thing about you can't just use brown and assume that brown means homozygous dominant, because it's both homozygous dominant and heterozygous in this case. Just give you stuff like this where it's, here's the frequency of something. And remember, if it tells you number of individuals and you need to find the frequency, you're just going to take number of individuals divided by total number of individuals, like number of brown butterflies divided by total number of brown butterflies. That would give you a frequency. If they're asking for the allele, it's different than the phenotype. So you'll just have to, figure out how to get from one to another. And there's like practice problems in Unit 1. So if you want to go download those practice problems again, I think I put them on the Miro board, but they're also in Unit 1 stuff too, definitely in the learning module at the very least. You can go do that and practice some of those if you need to. I just got a message from somebody. Let me just make sure it's not someone who's having trouble getting on the board or something. You will need to know some dates. I think they're all listed with the dates you need to know are listed in the, in this PDF thing. So chapter 23, on that page of chapter 23, I have like the specific times those individual events happen, the specific, billions of years ago or millions of years ago. And then obviously what order they're in. So I do have some ordering questions, like here's some events, put them in order. And do recall that plants and fungi move to land first, and then animals can move to land, because, in large part because the fungi and plants provided things, especially fungi help plants grow, plants allowed the animals to have something to eat there. And then they could also, of course, eat each other, and then the pollination and all that kind of stuff, because the insects move first to land. Then later we get like 100 million years later, we get the vertebrates moving to land as animals, but plants are first. So, it doesn't specifically say that here. It says fungi, plants, and animals. Fungi and plants are first, and then animals are second. But those would be the only real dates you'd need to know. Origin of Earth, we can write these on here. 4.6 billion years. Prokaryotes arise sometime between 3.5 and 3.9 billion years. And I'm not going to be crazy or something and say like, is it 3.2 or 3.5? Like, it will be obvious. Do make sure, though, you read whether it says billion years or million years, because those are different, right? 5 billion years and 5 million years. Obviously, it's 1,000 times different. Oxygen revolution. Oh, that's 2.5-ish. 2.5 billion years ago. So that's photosynthesis. And that's who did photosynthesis first? What type of... Well, who did oxygenic photosynthesis? I should specify that. What kind of organisms?
Cyanobacteria.
Yeah. Good job. Cyanobacteria. Now, cyanobacteria did exist earlier. Like the stromatolites were cyanobacteria fossils. They did exist earlier, but they just... we know that they didn't grow like super... extensively until the oxygen concentrations start changing, and we can tell that because like the metal deposits are changing in the earth. So clearly oxygen levels are different because you see the oxidation of the metals in different forms and things like that. So yes, okay, cyanobacteria, good. So we have those, but those are the ones doing photosynthesis first. Eukaryotes arise about like 1.8 billion years ago. Land plants and fungi. I guess like 500 to 600 million years ago? Now we're a million years ago. Animals are coming around that same time. I do have a picture here. Let's look at this in the slides. So this is on the It's after the Hardy-Weinberg slides. It's the one, I guess it's the fourth row down. This timeline here. Oh, I guess I should have just looked here. Starts at the bottom, 4.6 billion years, age of the Earth. This is way better. Way better. All right. Then we've got the signatures of life, 3.8. First fossils, 3.5. Which are prokaryote fossils. Those are the stromatolites. Here's the oxygenation. 2.4 is what they're saying here. So 2.5 is what I said. Because I just remember it by like, okay, 4.6 to 3.5 it's like, that's about a billion years. So it's like Earth forms, a billion years, life happens. I know that's prokaryotes. Then I know it's about another billion years for the oxygen thing, right? 3.8, 3.5 to like 2.4. It's about a billion. So it's like a billion to life, billion to oxygen, another almost billion to, or half, I should say, to eukaryotes. So eukaryotes are 1.8 billion. So it's about 2 billion from prokaryotes to eukaryotes. Earth is 1 billion to prokaryotes, prokaryotes to eukaryotes. It's about 2 billion. And then we have the here, land plants. Not 600. Oh, what are they putting it at? 525 or something? I should look that up. And then we've got vertebrates, and we have earlier than that we have insects though. I'm sorry, I should look those actual numbers up for you. It's not very helpful. But the order though, so all those things that were happening, the land plants, vertebrates, all moving are around 500 million years. You just know 500 million years, that will be sufficient for kind of all that stuff. Oh, and then Homo sapiens, we're looking at, probably 300,000 years for modern day humans. And that's modern day humans. Even the genus Homo, which is older than that, is still right at this very tip, because everything else is in millions of years and billions of years. So that's not even genus Homo. This is Homo sapiens. And then down at the end of this row, just a reminder about what Darwin never knew ideas, which go along with, obviously how evolution works, in large part natural selection. So here's some little videos, and that might be helpful. This figure that was from your notes from what Darwin never knew. So just to remember that sequence of events here, we've got the body plan genes that code for protein transcription factors, which bind to switches. Those switches regulate other genes being expressed. They call those stuff genes. Technically, they could also regulate other body plan genes. So it's this cascade of transcription factors being produced in certain areas of the body, those turning on certain genes in certain regions, and those genes then producing certain proteins that do stuff and make the body different ways in different locations and do different chemical reactions in different locations, that sort of thing. And so I have that kind of written here. And this is the most common way of, yes, you can have, like the rock pocket mice. Yes, you can have genetic changes, mutations in the genes themselves. So rock pocket mouse has a genetic mutation in the MC1R gene, which codes for the receptor. It is a different form of the receptor that changes how dark or light the coat color is, because it changes how well it's suppressed for making eumelanin, the pigment. Yes, you can change body plan genes, but a lot of times mutations in the actual gene themselves, whether it's the stuff gene or the body plan gene, often kills the organism. Because without without making any functional or normally functioning version of the protein, the organism can't develop. So what happens here is that a lot of times it's these switch regions, the regulatory regions, where the mutations, they arise everywhere. But where do they actually make it through is the switches, because the switches often will just change gene expression in one place, but not another. So that example here was the stickleback thing. This is the staining. So you can see this is staining for PIDX1 is the gene. That's stained in this blue-purple color. But you can see here the blue-purple is expressed in a number of different places on the left in this fish over here. It's like a fish embryo. And it's expressed in a number of spots here. And then you can see on the right side, it's no longer expressed in those pelvic fins, but it is still expressed in the mouth. If we looked, it is still expressed in the brain. In the pituitary gland. So it turns out if you mess with the PIDX1 gene, kills the embryos, they'll never develop, largely because they have to have it in the brain in order to live. So if it's a change in the switch though, and it only changes the switch that turns it on in the pelvis, then it doesn't kill the stickleback. It just means that it doesn't have pelvic spines. So the switches is the most common way that we get actual genetic mutation making a difference in and then like actually leading to natural selection. Okay, so unit 2 was prokaryotes and protists. So that talked about the origin of life, like origin of cells. Talked about the endosymbiosis idea, because that's part of the origin of eukaryotic cells. We talked about the characteristics of bacteria and archaea and the types of groups there. And we talked about the characteristics of a bunch of different protist groups. For all of that, I would give you characteristics and expect you to know what group it's referring to. So there's a bunch of boards to the right called review practice boards. It's like, I don't know, 17 of them.
It is these, okay. So are they just like all identical for us to all use?
They are, so everyone can have their own to practice, to like work on. So one thing you'll notice on here is just an example of like a concept map I made about microevolution. It has a bunch of blanks in it, so you could try to fill that out, and then you can see like, do you understand how all the ideas connect together? I do have a key I can put on here that I can you can look at after you see if you can fill it out. So that's one thing that's on the board, is this microevolution. Of course, you can make concept maps by hand or on Miro or another program, whatever, for any topic you want to. This is just an example of microevolution, which is a lot of the stuff that we just talked about. But that's just an example of a study tool. There's only certain groups. We can go over some of the characteristics in a minute. Let's look at the groups though, because I know there's a lot of different groups. I know the prokaryotes and protists are unfamiliar, unlike things like an earthworm. Some of the animal groups are unfamiliar, but they're definitely more familiar than the other, unit 2 stuff. So you'll notice here on the left side, there is a chart here that has the stickies at the top, bacterium, protist, and fact. They're just different colors. Those are that's just the key for what the colors of the stickies on the left means. Okay, so you'll see the stickies on the left say Staphylococcus aureus, Clostridium botulinum, Giardia, Bacillus anthracis, etc. These are all going to be disease-causing ones. So as far as like what this is going to show you the kinds of ones that specifically, the specific names. If you need to know them specifically, they'll be on that PDF review sheet and then also here. So just take a moment and see how much you can remember about the groups that cause these different infections, and then there's a fact about them. So whether it's the vector that transmits them in yellow, or something else about how they're like what the kind of disease is, or different strains of them, that sort of thing. So see if you can fill that out. Let's just start. I'm just going to start with Giardia. I think most people had that at the beginning. So Giardia is a protist, in other words, a eukaryotic organism. So it's a eukaryotic single-celled organism that causes an intestine infection called giardiasis. And there's 2 of them, interestingly, that have cysts in water. Cysts from feces in streams, cysts from feces in water. Those seem pretty similar. So how are you going to know the difference here? Well, one has information about that it has lobe pseudopodia. So I don't know if you remember Giardia, when we looked at them, they were a little hard to see, but they had flagella instead, and undulating membrane kind of thing. So these are going to be from cysts in streams predominantly. You can get it other places, but predominantly streams. So you can kill them, these guys, by boiling your water. These are the ones where you're hiking. You don't want to just drink water out of the stream, even if it looks pristine, because it could have, often has Giardia cysts in it. And that is a very common intestinal infection, which I did find out that is not commonly tested for. So I've known a few people who've had some terrible intestinal infection, and then no one could figure out what it is, and they finally test for Giardia, and turns out that's what it is. So if that happens to you and no one's figuring it out, you might mention, hey, can we test for Giardia? All right, sleeping sickness. So Trypanosoma causes sleeping sickness. You'll notice it's on here twice because there's multiple species that cause different things. One of them, sleeping sickness. What goes with sleeping sickness? Who passes sleeping sickness on? Yeah, okay. Causing sleeping sickness passed by the tsetse fly. Then there's another species of Trypanosoma that causes Chagas disease, and that one is passed by this kissing bug or triatomine insect. That one they have in Central America, South America, I think maybe Mexico as well. So it's right near us. Trypanosoma brucei is sleeping sickness. Trypanosoma cruzi is the Chagas. I won't make you know the difference between the two. Trypanosoma, if you know it goes with those two, that's good. Okay, malaria. I saw most people did have this one. So malaria is also caused by a protist called Plasmodium. There are a few species. Plasmodium vivax is often what you hear, but Plasmodium, the genus, is enough. Not all Plasmodium causes malaria, but if you know Plasmodium, malaria, and who passes that one? The Anopheles mosquito. I even saw some people who knew malaria was passed by the Anopheles mosquito but weren't sure what the protist was. So that's good. You have that side at least. But yeah, Plasmodium is the protist that the mosquito is passing from person to person. Which has the vectors, of course, remember, have the part of the life cycle inside them, but it doesn't bother them or kill them or make them sick. And then part of the life cycle has to happen in the host, one or more hosts, and then gets back to the vector, finishes the life cycle, etc. Neuroparalysis, called botulism. So that's going to be your Clostridium botulinum. That's a bacteria. See how it's in blue? Clostridium botulinum causes neuroparalysis, specifically botulism type neuroparalysis. That one you can get from canned food. That's why you don't feed honey to infants, because there's sometimes small amounts of botulinum toxin in there. It's fine for adults, but for little ones, it can be too much for their bodies, their immune systems. We could also add here, if you want to put another sticky, this one's also used in Botox. I mean, it's a derivative. It's not the original. It's not like they extract it from the bacteria itself, but they have a variant of the neurotoxin, the botulinum toxin, that's used for Botox and other similar neuroparalytics, Dysport. I don't know, there's some other one I can't remember the name of, but that's both for cosmetic purposes and for other medical things like excess sweating, migraines. That's also why you don't want to eat the food in a can that has puff on it. It's not good because clearly something was growing in there making gases that you don't want to eat. Okay, Clostridium tetani. Did I put that in? Maybe someone else did for me. Clostridium tetani. Yep, neuroparalysis tetanus, similar type of thing. And that one is entering through the wounds. So that's why a lot of times when you get cut and you've been exposed to like water and things like that, we're not sure how clean it is. You want to get a tetanus shot because you don't want to have neuroparalysis and not be able to breathe and die from suffocation. Because your diaphragm is a muscle. Okay, Lyme disease. We're back or onto bacteria. So Borrelia burgdorferi is what causes Lyme disease. Oh, that one is not italicized. It should be. All these should be italicized. Borrelia burgdorferi causes Lyme disease. It is passed by deer ticks. Let me move these down. Then we have staph infection. So I saw a lot of people use clues for this one. Staphylococcus aureus causes staph infection. There are many types of bacteria with staph in it, right? Because that word just means like clusters, like the colonies are in clusters. Not colonies, the cells. But so it's not like you see staphylo and you're like, oh, staph infection. But this one is the one. Staphylococcus aureus causes staph infection. And that one has a drug-resistant strain called MRSA. Sometimes people call it MRSA. So one you've got to be really careful about not getting, because then most of the drugs aren't effective on it because of natural selection. Anthrax poisoning. This name, Bacillus anthracis. Anthracis. That's where the name comes from. It is going to be one that forms resistant endospores. It is found naturally in the soil, not in large amounts, but it has those endospores, which means that I'm on the first board, and I'm on the first board. I put a star at the top. You can pick a different one though. Yes, so it forms resistant endospores, which means that, and this is actually true of Clostridium as well. The Clostridiums form resistant endospores. So, that's why you can't just boil the food that's puffed up to make sure it's okay. Like, you can't just cook the food and expect it not to have botulism poisoning in it still. So the anthrax is the same way. It forms these endospores, which then you can breathe in and get really sick, and even die from that. There's 2 STIs in here. Actually, 3 STIs in here. There we go. Gonorrhea, chlamydia, and Trichomonas vaginalis. So how are we going to know? Well, this first one, permanent female reproductive system damage, can cause blindness in babies if they're infected during birth. That one is your chlamydia. I mean, babies can get gonorrhea, I think, eye infections, but chlamydia is the ones that's classically known for the eye damage, which is why they always do STI testing before in pregnant mothers, because it's really important to keep for baby health. It can really damage them. I made a little mistake in this part in the original table, so I'm going to fix it here. So gonorrhea is just the infection caused by the bacterium Neisseria gonorrhoeae. You can see that last part of the species name matches gonorrhea. So I had originally that gonorrhea was the genus, but that's not correct. It is an STI, and there are multi-drug resistant strains spreading, which means they're not able to be treated by many or any drugs. And you read about this in your CDC antibiotic resistance quiz during the semester, during Unit 2, that was I think it's one of the top 3 that they're really concerned about this antibiotic resistance in that species. Correct condom usage can really help cut down on the spread of this infection, which means that we won't have to use as much antibiotic treating people, which means you won't have the selection for those resistant strains. So that's something really important. Entamoeba is, in fact, the other intestinal infection. Cyst from feces in water has those lobe-like pseudopodia. Entamoeba is one of those amoebozoan groups in the Uniconta supergroup. And then Trichomonas vaginalis is the last STI here. I don't have anything specific about this here. Forms resistant endospores. Oh, I did have this. That's interesting. Okay, so that goes with Clostridium tetani, and that also goes with Clostridium botulinum. Then we have our groups that are the different types of protist groups that are not specific species usually. So that's these little Venn diagrammy things that I've made in the next column. So there's the first little Venn diagram is looking at algae cellularity. And so here's the different types of algae. So bigger algae, seaweed algae, small algae, like unicellular, stuff like that. So we can sort those in there. And of course, when the 2 boxes overlap, that means that they could, in different species within that group, could both be unicellular and multicellular. So in this orangey box, that's the pink and the yellow overlapping. That orangey box means that group could be multicellular or colonial. The purple box means that group has some species that are colonial and some that are unicellular. So we can sort those algae. So it's going to basically be I give you descriptions, and the descriptions may include the cell wall material. They may include whether they have pseudopodia, cilia, or flagella, which is on here. They may include if they're unicellular or multicellular. They may include even economic things like, oh, they're used for scrubbing agents, or they're used in the fossil record, you know, because we talked about that a lot with the different types of protists. I also did put on here that that Excel file that you had the protist organization thing. So if you wanted to download that again, if you don't have that, it's on everybody's boards, but that's what this little file is that's right next to the algae cellularity table. So it doesn't make much sense if you look at it in here. But if you wanted to download that, you have one in your board, you could do that too. Remember, protist is like, you know, a catch-all term for eukaryote that's not a plant, animal, or fungus. Some are more closely related to animals, some are more closely related to plants, some are more closely related to fungi. They don't really have any consistent characteristics except for being eukaryotic. So, but it's a useful term. Not even all of them are unicellular, right? There's multicellular, there's colonial, they're all over the place. So there is no synapomorphy of a protist. John Paul, are you going to say something?
Can you explain the algae pigments? I don't even remember this.
Oh, you don't remember
Except
this.
for chlorophyll A.
Except for chlorophyll A. Okay, fair enough. Yes. So these are useful characteristics to be able to identify the different groups because they have the different pigments. And actually, if you look in the little file, the Excel file, if you end up downloading that, there's a drop-down menu thing for different pigments for different groups. Clues in the names. Chloro is actually meaning green. So chlorophytes, charophytes, well, particularly chlorophytes. Chlorophytes is referring to, the chloro word actually means green, like chloroplast, chlorophyll, all that kind of thing. So the chlorophytes are going to have chlorophyll A. They don't have any of the other ones. They just have chlorophyll A. And they are type of green algae. So if we wanted to add that to there, we could. So chlorophytes are a type of green algae. They've got chlorophyll A in it. They got it from their endosymbiosis, the primary endosymbiosis of that chloroplast. The charophytes are also a type of green algae. So they are more closely related to plant green algae, but the charophytes are also green algae. Green algae also has chlorophyll A. So green algae is going to have chlorophyll A's. Plants have chlorophyll A as well. Okay, then we go to red algae and brown algae. So red algae can grow quite deep because of the pigments that it has, and that's why it looks like a red color. So red color means it's not absorbing the red, which means it's absorbing the other stuff, which includes blue light. Blue light's a really high energy light, like fast wavelength, you know, or short wavelength, high energy, so it can penetrate quite deep in the water, which is why you can get red algae kind of living relatively deep, because that light can get all the way down to them and they can absorb that blue light pretty well. You might know erythro, like erythrocyte means red blood cells. Erythemia means your skin's all red. So the erythro thing is referring to red. That's actually where that word is coming from. Cyano is referring to blue. So both of these phycocyanin and phycoerythrin are the red algae pigments. And that's important because it's what allows red algae, especially the phycoerythrin, but also the phycocyanin. The phycoerythrin, though, allows them to live quite deep. They don't have to, because they can also absorb light near the surface, especially with the phycocyanin, because if it's cyan, that means it's reflecting like blue light, which means it's absorbing the red. So it has the ability to absorb lots of wavelengths, allowing these red algae to live at a lot of different depths. In the review session, we talked about this in a few minutes, but I thought I would just insert it here so it's all together. In addition to the phycoerythrin and phycocyanin, red algae also has chlorophyll in it, so it has all three of those pigments. Brown algae, fucoxanthin, that's the brown pigment there, it's like olivey kind of brown, and brown algae has the fucoxanthin pigment, which is does not absorb light as deep. It's more absorbing, like not a lot of the blue light, you know, but it allows those guys to only live more near the surface. So brown algae is going to be found more near the surface. Can be quite large, like kelp forests, but they're not going to be really deep in the water like you might get with the red algae. And just to clarify again, which we discussed later during the review, though, that brown algae has fucoxanthin in it, but it also has the chlorophyll in it as well. Now, interestingly, brown algae came from the secondary endosymbiosis of a red alga. So we might think, well, why doesn't it have the phycoerythrin and phycocyanin? But from what I've researched, that the ancestral red algae likely didn't have the actual entire, genes that coded for the phycoerythrin or phycocyanin yet. Those developed like when the red algae was doing its own thing after the endosymbiosis event, the secondary endosymbiosis event that led to the brown algae. I do want to draw a phylogeny, though, just to remind you about this primary secondary endosymbiosis. 3 domains of life. The most divergent from the rest of them, which is the first to diverge, bacteria, one of the prokaryotic groups. Do make sure you know the characteristics of bacteria, like what those things are. What's a plasmid? What are the fimbriae? What's the capsule? The terminology of the parts of that. We already talked about the gene transfer, but like what's pilus? What is, like, they have no nuclei or no nuclear membrane because, that's why we consider them prokaryotic. No complex membrane-bound organelles inside, maybe simple compartments. Can have highly folded membranes, though, like especially cyanobacteria. That's how they're going to do the photosynthesis and such. Yeah, so all those kinds of things. So if I describe a bacterium, then I ask, like, what feature is allowing it to do a certain behavior? For example, so this bacteria is not susceptible to drugs that prevent polymerization of the peptidoglycan wall. What kind of cell wall does it have? And you kind of remember, there's those gram-positive, gram-negative, and then there's other. But if we're thinking gram-positive, gram-positive have lots of peptidoglycan on them. That's why they retain that purple color and they stain purple. If it has lots of peptidoglycan, gram-positive, then it would be susceptible to drugs that basically prevent the peptidoglycan from holding itself together or from being built. So those would be susceptible to the drugs. So peptidoglycan, they're staining purple, those gram-positive. Gram-negative has a thin layer of peptidoglycan with that layer on the outside of membrane, like lipopolysaccharide membrane. So they're not resistant because they're not susceptible because they, it's not like they're genetically resistant. It's just that their structure is not based on having a thick peptidoglycan layer. So the drugs don't affect it. Plus that membrane on the outside of the cell wall, the second part of the cell wall, that membrane part, prevents that drug from getting in there too. So they stain pink, gram-negative, because they don't retain that purple because they're thin peptidoglycan layer. So if I ask you something about them moving around, you'd probably say, well, they probably have flagella or flagellum, flagella, singular or plural. If I ask you something about them sticking to a surface, and that could be a few things. Fimbriae are little kind of projections that help them stick. A capsule is a sticky coat that helps them stick, also helps them be not detected by the immune system. So just do make sure you go over those terms. Okay, so bacteria. Then we have our groups here. So this is just going to be an endosymbiosis kind of thing. There's the archaea group. We didn't do too much about that. Methanogens, what they're like. Those are archaea, the archaea are more closely related to eukaryotes. Some of them live in extreme environments, and maybe you'd want to know what a halophile is and a thermophile, but I don't have massive detail about archaea, besides understanding where they go in the phylogeny. And then we have this group here, which is the ones we've talked about for most of this semester, called Eukarya. And we have those supergroups of Eukarya. I talked about 4-ish, although we said one of them is a little bit defunct because it's in a number of other groups. I guess we also talked about haptophytes and stuff like that, which are other groups. So we had a little bit bigger than just 4 supergroups. Okay, so we have an event here, one endosymbiotic event where you have the engulfing and not digesting, right? That's what endosymbiosis is. Engulf, don't digest, exchange genetic material so that you become dependent on each other. Engulf a bacterium in this case. So that's primary. Primary is engulfing bacteria. So we've got this primary endosymbiosis event of that alpha proteobacterium. And that's going to turn into the thing that all eukaryotes have, all eukaryotic cells have, unless they are degenerate or maybe they've lost them, but we see evidence in all the cells. What did that turn into there? What organelle? What organelle did the first endosymbiosis?
Mitochondria.
There you go, mitochondria. All right, so alpha proteobacterium ended up becoming a mitochondrion. We know that's the first one because all the eukarya have it. Then we have a second primary endosymbiosis event, and then there's actually a few more. I also just saw a paper about another primary endosymbiosis event. They found a type of coccolithophore. It's a haptophyte coccolithophore. That's the ones we talked about, made the White Cliffs of Dover and stuff. They found a species that actually can do nitrogen fixation, which is the only eukaryotic species they've ever found. Nitrogen fixation is a bacteria thing. It's like bacteria thing. And so what they've done is they've actually found that it is from, it's good evidence that it's from an endosymbiosis, a primary endosymbiosis of a nitrogen-fixing bacterium that then became dependent on the host cell and the host cell on it. And so now they have an example of a eukaryote that can fix nitrogen. It's unusual. It's the first one. This just came out last month, a couple weeks ago. So that's another primary endosymbiosis event of a nitrogen-fixing bacteria. That would be in the haptophyte. Just one species, though. Anyway, so here's another primary. Let's do this here. Let's do this here because this is easy. Okay, so now we have another primary of a cyanobacterium. And that forms the chloroplast. What lineage is going to come from this, that's just going to arise from this chloroplast, from this primary endosymbiosis, the cyanobacteria? Who should I put on this top line up here?
Archaeplastida?
Archaeplastida, good, because it means ancient archae, ancient plastid, meaning ancient chloroplast. So it's the one that got the chloroplast from the first time chloroplast arose. That's how you can remember that. Good. Archaeplastida group. That's the group with the glaucophytes, the red algae, the green algae, and the land plants, or embryophytes, which then you'll branch out to there too. Then we've got these other events that are secondary endosymbiosis events in some of the groups. And they come from the red algae and the green algae in this Archaeplastida lineage getting engulfed. So it would be later in time, and this happens many times, multiple times, but there's two. It's not in all the groups, though, in here. So this is secondary endosymbiosis of, let's say this is going to be of a red alga on this bottom line and of a green alga on this top line. And like I said, though, this is like a very crude drawing, right? Because it's not like everybody in the lineage gets the secondary endosymbiosis from the same event. Let me write that here. Secondary endosymbiosis happened many times. And gave rise to many eukaryotic groups. It's not once per supergroup. Which is what it looks like here. The primaries are once. These other ones are not. But the secondary endosymbiosis for green algae, that's going to lead to groups like the euglenids. They came from secondary endosymbiosis of a green algae. Chlorarachniophytes. They're not all in the same group. This happened a number of times. Some will say, for example, euglenids. Another time that this happened was a number of times in the Sarclade. That was all with red algae. And then the primary endosymbiosis in that one special group, we're not worrying about that. And the green algae and the chloracneophytes, those are in Sarclade too. For example, diatoms had this red algae thing. Then we have another group here that doesn't have any primary or secondary endosymbiosis events. That's why I've drawn it separately here. That's the Uniconta. Uniconta didn't have any of those. That's the lineage with the amoebozoans. That's the lineage with the pisticons, which are include the fungi and animals. So no secondary endosymbiosis events. So what kind of groups came from secondary endosymbiosis? These are examples. We have the dinoflagellates, some of which have lost their chloroplasts now, but many dinoflagellates, they still have them. So dinoflagellates, remember those are the ones that cause red tide. Usually, they're also the ones that are coral symbionts. They've got cellulose cell walls. They're algae, so they do have the pigments in them. Let's see, diatoms. Those are the ones with hydrated silica cell walls. Also algae. Really strong, good for scrubbing products, help calibrate lasers, can absorb liquids, can also block the little, tracheal tubes of insects to kill them in gardening, that kind of thing. Who else do we have? The coccolithophores, which we've talked about. I just mentioned euglenids. Chlorarachneophytes. Didn't talk about that too much, but that one's one of those ones that had like the nucleus still in the chloroplast, like a little nucleus remnant. Good evidence there that it used to be a chloroplast, or that it used to be a eukaryote itself, because it had a nucleus, so it must have been a eukaryote at some time. Now it's a chloroplast. That's because it was an algae that got engulfed. Brown algae. Who's not going to be in this group? Green algae and red algae, though, right? Because those are from the lineage that was just from that primary endosymbiosis, not these guys. These are just some, these are some examples there. Leave it at that. All right. So there will be something along the lines of this, showing primaries and secondaries. You'll have to identify primary and secondary, what got engulfed, the different groups, that kind of thing. Okay. Should we do, I know it's getting close to time. I think we should do, the life cycles. So I have, and you can go on your board for this if you want to, or you could do your board later and see if you can fill it out. I don't have labels on here, so you'll have to make stickies, which I thought was going to be, I thought that would be easy enough. And I've made very simplified little life cycles for you. All right, so this first one on the top left, so we could watch and then you could make it later, is a plant life cycle. So we've got the two, remember it's alternation of generation, so we've got the two forms, like the two versions of the plant. Sometimes they're like inside each other. Let me go grab I guess most of you are over by me. Is that true? No, sorry. Oh no. Yes, kind of. Okay, one second. Let me go grab a picture to show you this. What was I doing? Oh, here. With this alternation of generations, if we look at the different plant groups, I just pulled this little thing over from the notes or made a copy of it. Sporophytes and gametophytes, those are the two kind of forms of the plants, or the two generations, but they're not always separate plants. So sometimes they are dependent on each other, and like they're two separate entities, like in the moss, let's say, or the hornworts, those bryophyte groups. Sometimes they're totally separate, like ferns are like that, other seedless vascular. Sometimes they're like the nesting inside each other, where it's like the plant that you see is the sporophyte, and then the little cells inside of the cone, or cells inside of the flower, or cells inside the pollen are the gametophytes. So as we, the seed plants, they're really, it's not really generation, meaning another organism. It's just inside of the actual plant. But we always have a sporophyte and gametophyte that are multicellular. So they're both multicellular stages of that plant's life, or parts of the plant that are important for reproduction. So except for moss, not just moss, but the non-vascular plants, which are the moss, the hornworts, and the liverworts, those guys are the ones where the green stuff you see is the gametophyte. Everybody else, the stuff you see, the plant you see, is the sporophyte. The flower plant, like the rose you see, is the sporophyte. The fern is the sporophyte. The horsetail is the sporophyte. The pine tree is the sporophyte, okay? So in this case, we're looking at a fern example here. So this is going to be the sporophyte. I will not force you to like figure out, oh, this is on the life cycle, I will have sporophyte filled in or gametophyte filled in, or maybe neither, but I'd have ploidy filled in. So you will be able to tell where to put it. You're not going to have to say, is that a picture of a fern? Which group is that in? I wouldn't do that. I'm not going to do that to you. But I would say sporophytes, you got to remember gametophytes or sporophytes, doesn't matter which one you remember because they're opposite ploides. I have memorized what sporophytes are. That's what I memorized first. And then they're diploid. So sporophytes are diploid, which kind of like makes sense because, you don't have to be diploid. There's no reason. Fungi are predominantly haploid, right? All fungal bodies, like the mycelium's haploid. Most of the fungus is haploid. We have other groups that are haploid as well. Bacteria are haploid. They've only got one chromosome, right? But sporophytes are diploid. So like I always think, oh yeah, the plant body that I see is the same as my body. So the tree I see is diploid just like me. And then if you know the tree is sporophyte, then you know it's diploid, or you can just memorize it. But in any case, sporophytes are diploid, which means the opposite of that, gametophytes, are going to be haploid. Because one generation is one generation is the other. Whether it's a single, they're multicellular. Both of these are multicellular as well. So the sporophyte, we like this, the sporophyte is going to make spores, because remember, phyte means plant or plant-like. So the sporophyte is the spore-producing plant. So these guys are spores. Now, spores, do you guys remember how we went over like the ploidy of a bunch of different stuff? What are what's the ploidy of spores, whether it's from fungi or from plants? Haploid or diploid?
Haploid.
Always haploid. Good. What else is always haploid? There are a number of answers.
Gametes.
Gametes are always haploid, eggs and sperm. Yep. The mycelium of a fungus is haploid, and gametophytes are haploid. So then we can always figure it out, right? If the spores are formed from sporophytes, spores are N, sporophytes are 2N. What had to happen between those two to make the gamete?
Meiosis.
Had to be meiosis because it halved the ploidy. Halved? Made the ploidy half of what it was. Good. Meiosis is what's forming spores in plants, not gametes. The spores need to grow up into a gametophyte because spores are single-celled, gametophytes are multicelled. Whether they're on their own or within the other plant, they're still multicellular entities. So N to N, The type of division to keep the ploidy the same, mitosis. Then the gametophytes are there. What are they going to make? Sporophytes make spores. Gametophytes make gametes. Thank you. Yes. Gametes. And you guys just told me that those are always N. So that means gametophytes are doing division to make gametes, N to N. So that must also be mitosis, same ploidy. Gametes come together and combine like the sperm donates the DNA to the egg, whether that sperm swims. So there's two groups in which it swims. It swims in the seedless vascular plants like ferns and horsetails and that sort of thing, lycophytes. It also swims in the bryophyte group, the non-vascular. So that's like the mosses, the hornworts, and the liverworts. That's why those are still kind of like need water more. That reminds me a lot of fish and amphibians. Amphibians in particular, right? Because these are all land groups. They're not like in the water still. Land plants, land animals, vertebrates. But they still have swimming sperm that are dependent on water. So that's interesting. So two gametes coming together though and combining. Fertilization. Fertilization always makes a zygote. If you had two N gametes combining their DNA, N plus N is 2N. So the zygotes are always 2N. And then you'll notice the zygote is a single cell. It grows up into a sporophyte. And plants do have an embryo and everything. They don't do gastrulation the same way and all that as animals, but they do have embryonic stages and all that. So if we have our zygote growing up into a sporophyte, we have 2N to 2N, which we have same ploidy, so mitosis. That's the plant life cycle. There, like I said, there's variations of where it happens and all that stuff, but and you can use that chart above, it's in the notes as well, to kind of show you where gametophytes and sporophytes are. I'm not going to do a thing where you have to identify, you know how like on the exam I had the thing where you had to look at the picture and identify the gametophyte and the sporophyte and the gamete and the spore, like within the pollen structure and within the ovary, like the ovary of the pistil or carpels. I'm not going to have anything that specific for you. I'd have a life cycle. I'd expect you to know mitosis, meiosis, fertilization stuff, and which features, like the features for each plant group. So it's like you're walking in the forest and you see a plant that has seeds and pollen, but no flowers. What have you found? You'd say gymnosperm. Or a plant that has no vasculature. You'd know that would be in the non-vascular plants, a bryophyte. So it's one of those fungi. It's either hornworts, liverworts, or mosses. If they had flowers or fruit, if they have fruit, they have flowers. If they have flowers, they necessarily have fruit. Those are in the angiosperm group. So I'll just do those kind of big groups, except for the phylogeny where you'll need to be able to put all those groups on the phylogeny like we did before.
Everything else, I think we can kind of go.
Yeah, I feel like animals we're pretty good on. For the phylogeny, for the animals, you just know what groups to put stuff in based off of a feature I give you. So like, for example, if I told you, because some of them are polytomies, right? Where it's all the lines coming off the same thing. So you're like, well, how do I know what order? If you remember on the exam, I would either give you an example, like say, octopus, and you'd know to put mollusk. Or I would do a thing where I would say like mantle is a feature, and then you'd know to put mollusk. So that's how you're going to know where to put them into a polytomy. Or I'd give you something like comb jelly, and you'd know to put ctenophore in there. So that's for those, that's the animal phylogeny. So just, make sure you know those features. I think you'll be okay on that. Fungus life cycle. A little bit about the fungal groups, I'm not going to have any pictures of the reproductive structures or anything like super specific like that, but you do want to know what's the feature of the basidiomycetes, having this basidium club-like, reproductive structures. Ascomycetes have the sac structures of the sac fungi. Zygomycetes have the zygote structures, and then like chytrids are the ones that infect the amphibian skin. They look similar to nuclearids, which are the protists. And then the glomeromycetes, they include the ones that are the mycorrhizal relationship. So those mycorrhizae, which are the roots and the fungi, the relationship of those together to do mutual exchange there. It's a mutualistic symbiotic relationship.
So we should be able to fill out the animal, plant, and fungal life cycles, right?
Yeah. So it basically be like the domains, and then you have the eukaryotic supergroups ones, specifically in those Archaeplastida and the branching of Archaeplastida, the branching of Uniconta, and then within those you'd have not the fungi branching. I don't need you to know that. Plant branching though, and animals. And then chordates within the animals. And they are, I'll make sure all of them are on here, but on your board, there are some blank phylogenies, which I can have a key to, which are also mostly from your book and such, which cover all the different groupings you need to know. Now, they could be written somewhat differently, right? They as long as the branching pattern is the same, so you may need to follow the branching. It might not just be like top to bottom is the same, right? Because it just depends on how do you branch up first or do you branch down first? That kind of thing. So yeah, but those are the ones.
Okay, so then if that's like we kind of have an idea on units 1 through 5. What's with the pseudo unit? Like, what do we need to know about?
Yeah. So for the only thing that will be about okay. So let me go. Ecology on the very last page of the PDF. And if you want to come over to me, you can on the board. But the PDF, that's like, all the questions or the topics to know. It says ecology, which is what you're talking about here. So the ecology ideas that you need to know are, okay, no worries about conservation biology because I elected not to do that in class. So you don't have to worry about conservation biology. You don't have to worry about, there's a few slides at the end of the, review slides or whatever. You don't have to worry about those. So the kind of ecology ideas, though, are basically a keystone predator or keystone species, like you did in your symbio. Just basic idea about that. And then ecology ideas that we've mostly covered in the other units. So chemical cycling, primary producer, secondary, primary producer, primary consumer, secondary consumer, decomposer, how those all relate to each other. I'd give you an example of some organism doing something. So it's a bird eating a worm. What's its role? And you would be like, oh, it's animal eating another animal. That is and what did that first animal, that's going to be a secondary or higher consumer, right? If it's a cricket eating a leaf, that is a primary consumer because it's eating the leaf, which is the primary producer. So that idea of roles in the environment. What are the other things? Let's see. Food webs, food chains. It's kind of all related to that. Oh, symbionts, which we've talked about a lot, like mutualistic, commensalistic, parasitic symbionts, how those, like I'd give you a scenario and ask you which one is it acting as, what kind of symbiont. Predator is usually referring to not necessarily animals. An organism that moves around to eat other organisms are predators. Herbivores are the ones that eat plants. Animals that eat plants. Yeah. That kind of stuff. So nothing super new. It's all like chapters 42 or 40 to 43 or whatever is at the end. I kind of integrated the most important parts as we went. So you're not going to need to know anything about population dynamics or distribution or carrying capacity or any of those kind of concepts. I do have on the slides at the end of the bacteria archaea section, there's a thing that says, it's like right after the oxygen revolution slide, 37.2, interspecific interactions are fundamental to community structure. You don't have to worry about 37.2. That's not relevant for you, but it does talk about competition, mutualism, predation, herbivory, parasites, and pathogens, the plus minus there, and some examples. And then this idea about logistic versus exponential growth, and then shows you this chemical cycling. So remember, energy goes through the system. It comes in through the sun initially, moves around by transfer of organic material, leaves as heat. Chemicals recycle because we can't create or destroy matter. So the chemicals just recycle from the abiotic environment into the organisms, and then back eventually from decomposers, break them back down into inorganic compounds. Oh, and I guess also the types of nutrition modes, that's sort of like a ecology thing. So photoautotroph, chemoautotroph, photoheterotroph, chemoheterotroph. Which ones use light, which ones use organic compounds, like that little chart, the diverse nutritional modes chart. So that's the only things from ecology ideas, stuff we've previously covered. Is that helpful?
So, yeah, yeah. So in general, would you say that I've just been kind of trying to figure out what would be like the best use of my time, because I'm going through like the final review. I didn't realize that you were going to have like a mirror board for the final, so that's really cool. But I was looking through the final review and I was kind of like, should I go through each individual old learning module, or like, because I'm trying to sort of, I basically like copy and pasted the review and was trying to like answer some of the questions and like study that way. But yeah, I guess I don't know.
I may also use the strategy of going back to your exams and seeing if there are any like easily identifiable topics that you're kind of like reading through it and you're like, oh man, I knew nothing about fill in the blank. Like turns out I hadn't learned anything about, organ systems of plants or something like that. The only thing, like I said, the only things about organ systems really are, like what does the leaf do? What does the stem do? That kind of thing. So that's also sometimes useful to see if there are a particular weak area that you have, and then you can go to the part of the review sheet about that, and then just study that part first. Because do you want to spend a lot of time on the evolution stuff if you feel really good about it? You might want to just skip to Unit 2 questions instead of starting at the beginning, because then you'll never get through them all. I mean, you might, but you're just spending less time thinking about it the longer you wait to do it. So I'd say maybe start with Unit 2. I think most people struggle the most with that. So maybe start with Unit 2.
For sure.
Get that underway. And this is always the case. I do really try, but Unit 2 is just so new for many people. And I do cut out a lot of it because you can learn so much about bacteria and microbiology and archaea and all that, so we don't go into massive detail. But that might be it. And there's a lot of good examples in the Unit 2 exam of like descriptors of stuff, and then you have to answer questions based off of that. So go back and look at those where it's like, here's a bacteria that has these features. Question. It's like that idea of being able to extract information, to answer a question about like what would their behavior be. If they don't have flagella, probably can't move. John Paul likes it. All right. So that's what I do. Maybe start at 2. Get those protists under your belt. There is some stuff about like with some animal groups to identify those, of course. And recall that all the vertebrates are in the phylum Chordata. And then all those groups underneath, Chondrichthyes, mammals or Mammalia, all those are subphyla or classes. The big one is Chordata. Unlike something like mollusks, that's a phylum. Chordata, that's a phylum. Arthropoda, that's a phylum. Sometimes people get confused though, and all of a sudden they're putting mammal or Mammalia as a phylum, because you're thinking, but that's one group down from that. So
yeah, I guess what I'm most worried about is keeping track of like all of the names of the groups and characteristics and examples for all of the different types of groups spanning across the units.
Yeah.
Just track everything,
Yeah, and let's see. I can pull over maybe some stuff we had on other boards that were like organizational kind of things. So that way you have it all on this one review board. Or of course you can copy and paste from another board too, because we had those animal charts that might be helpful to have on here, or a simplified version of that. Yeah, because you won't have to know everything. So maybe a simplified version of the animal chart. I could try to make that and leave a copy. I like leave a table up there. People can copy into their board if they want to. That kind of thing. Yeah. Okay. So let's do the plant. I mean, let's do the fungus thing. I understand if you have to go. It's getting late there. Fungus thing, and then we'll be done. All right. We're going to start. I'm going to start in the middle because that's the most of the time the fungus is existing as a mycelium. I like this response thing you guys are doing. Makes me feel like people are listening. I like it. Okay, mycelium. Always haploid, right? Most of the time the fungus is haploid. Mushroom structures, interestingly though, we shall get to those, are not haploid. I know that's what we usually see, but they're the weird stage, if you will, the fungus special stage. So we start with the mycelium. Let's go to the left. That is going to be our simplest reproduction, often what's happening, asexual reproduction, just where it's not splitting itself or anything. It's making spores. The spores go disperse. They grow up into a new fungus, or down, probably, into a new fungus. But that fungus is identical to the initial fungus. So new fungus is formed, or many new fungi, but each one's identical. So we start with the mycelium. We're going to form these little structures here, which are spores. Because pretty much anything that's formed in fungus is going to be a spore. I mean, almost anything. But it's pretty much going to be lots of spores. There's no gametes, remember? And there's no fertilization. People get confused about that. So no gametes in fungi. But there are spores. So if spores we know are N. So it's a mycelium producing spores. Oh, and it's through a spore producing structure. So basically like some part of the fungus needs to differentiate, or it needs to like develop into a spore producing structure, like a sporangium, like those lollipop things that we saw in the rhizopus fungus, the black bread mold fungus. Okay, spore producing structure makes spores end to end. So that process is going to be mitosis, right? Same ploidy. Right. And then the spores are going to have to grow up into a new mycelium. My picture is not exactly like this. I have a different version of the picture. But it's going to grow up into a mycelium end to end. More mitosis. I can just leave that. They call it germination sometimes. The spores germinate into a new fungus. You hear that with seeds as well. Seeds can germinate. It just means grow into a new one. But the spores are going to do mitosis to form a new mycelium. That's asexual reproduction. There's also asexual reproduction in fungi of budding. You know, the yeast can like literally just split themselves or split a small part off of the cell. So basically mitosis. But that's because they're unicellular fungi. Most fungi are multicellular, except for yeasts, which fall into a number of, well, mostly they're ascomycetes, but varied ascomycetes. Okay. Sexual reproduction, mycelium. This is when now you see there's a second mycelium coming in up here. So this is why it's sexual, because there's two mycelia that are going to contribute the DNA. That, you know, when you're doing sexual reproduction, you need DNA from two sources, whether it's the same individual or not. So even in a plant that self-fertilizes, that's still sexual reproduction. If it's, in that case, egg and sperm making an offspring. In a hermaphroditic animal that does sexual reproduction, if it self-fertilizes, it's still sexual reproduction because it's egg and sperm. Here, it's two different mycelium. They're from two different fungi in this case, but because you have two inputs of genetic material that's different, that's sexual reproduction. So mycelium come together, and we do a process called fusion of the cells. So the ogamy part, right? Or gamete, I guess it's really the gamete is the marriage. The plasmo is the cell. So first we marry the cells together, basically just fuse two cells. So that's where it's called the plasmogamy. Plasmogamy. Cell marriage. So now you're sitting there in this sort of like intermediate. So unlike fertilization, where really the sperm and the egg don't fuse, at least not in animals. I don't think they do. No, they don't in plants either. Plants just send their nuclei down the pollen tube. So it's just nuclei. So all the sperm gives to the egg in plants and animals is the nucleus. So then the nuclei just fuse right away. In this case, the cells are actually fusing. So the nuclei just hang out by themselves for quite some time. Could be a very long time. It's still until a trigger causes the next thing. So at this point, they're called the heterokaryotic stage. Hetero meaning not multiple, that's the wrong word. Opposite, different. There we go. Hetero meaning different, karyo meaning nuclei. So we're in a stage here where it has many different nuclei. It could be just two nuclei, or there are cases where the mycelia have a lot of nuclei in each of them. So when they fuse, they're just like a bunch of nuclei in the one cell. So heterokaryotic. Basically, we can represent that by saying N plus N. All that means is it's going to be a two or more haploid nuclei. Half of them are from one parent or one initial cell, half are from the other. If we just want to think about two, we just have one from one from the other. The cells fused, hey, they're hanging out, haploid nuclei. Then you go to the a trigger has to happen. So this is actually the fungus, the mushroom. Mushrooms that you see are heterokaryotic. So if you looked at the cells inside a mushroom, not under the gills, because that's onto the reproduction, but the rest of the mushroom body, it's gonna all be cells with two or more nuclei in them each that are both haploid. So what are we going to do next? Now there's some trigger in the environment, probably favorable conditions. That's going to cause now the nuclei to pair up and fuse. So there's always just two that fuse, one from one initial parent, the other from the other. That is called the marriage of the nuclei, which is karyogamy. The gamete again for marriage, and the karyo for the nuclear marriage. Maybe that's what this is. Sure. That's the karyogamy. It's interesting too, wherever a mushroom's growing, that means, because it's forming for sexual reproduction, that means that two fungi connected, and they've combined the cells and it's growing into the mushroom, and then sexual reproduction is happening under the gills. And by sexual reproduction, I mean these last, the karyogamy and then the zygote and all that kind of spore formation. But yeah, that shows you the tips of where, or the intersection of two fungi in your yard. Okay, so now we have the nuclei together, and that is forming a zygote. So even though it's not from egg and sperm, still called a zygote. 2N, still one cell. And then that zygote needs to grow, it can't grow up directly. This is different, right? Zygotes we often think of as growing up directly into the new organism, like goes through an embryo, but fungi don't do that. They actually, the zygote is the only diploid stage, and it's going to actually become not a zygote very rapidly. So the zygote is going to need to make some spores, because the spores are what can form a mycelium. Zygotes can't form mycelium. So what we're going to have to do is make a spore-producing structure, kind of like the zygote. It's not going to divide into it or anything. It's usually just in that cell, it's going to be able to do the spore production. So the zygote is going to be able to produce the spore-producing structure. So that's going to make the spores. So these are. So the spore-producing structure has made spores, and it went from zygote of N to spores, zygote is 2N, spores is N. What did we have to do to make spores? Half the amount of DNA, right? That's meiosis. So within the spore-producing structure, which is part of the zygote, spores are made. Spore-producing structures are the things like the sacs. I don't know if you remember when we looked at ASCO, we looked at Paziza, had all those little finger sac things all lined up in a row, and then they had those dots inside of them that were the spores. Those sacs are the spore-producing structures. We also looked at the ball, that really spiky thing that looked like a kind of like a gumball from a tree, but that was from the Rhizopus solanifer. That was the zygotes inside of that spore-producing structure. It forms the spore-producing mitosporangium and then, or myosporangium, and then makes the spores. So it's by meiosis. I'm not going to have you identify any of those specifically. I'm just trying to get it in your mind. So zygote, 2N, spores, and meiosis there. And then these little guys got to grow up into a new mycelium because it goes from single cell to multicellular. And every spore is going to do that by mitosis. Most of them will die, but if they're favorable. We can note, though, genetically, these spores are different, right? The spores are genetically unique in sexual reproduction, where they'd be genetically identical in asexual reproduction. So you just have a bunch of identical spores in asexual, but every spore that's formed by meiosis is unique in sexual reproduction.
So I guess I should just ask for the sake of just reminding everyone. So we're taking the test in person for once, and it's gonna be 8 a.m. Tuesday, and it'll take what, 2 to 3 hours?
Yeah, I think because some people have class right after. I've looked at, not right after. I've looked at when the next time is that would be possible for people to have an exam, and I think it is. Does anyone have an exam right afterwards that's here? I have looked. No. 8. I think I gave you two hours to take the exam, 2.5 hours, something like that.
Yeah, on the online thing it says 2.5 or 2:40 or something like that.
Yeah, it'll probably take 2. I went through the exam today just, to try to check everything. It took me 40 minutes. But I made the exam. So, but I wanted to make sure I actually went through and did everything. I was curious how long it would take me because, even if you know everything, you still do need to read every answer, because it's not like I know every answer just glancing at it. I haven't memorized every question. And then I also wanted to make sure, because some of them are a little finagly back and forth with the phylogenies and stuff. So you have to write down your things and then transfer the answers. So yeah, it took me 40 minutes, which means probably takes you guys about double, I would say.
Okay, and then we're allowed to have a 3x5 note card.
You can have a 4x6 note card for the final.
Oh,
okay. There you go. Same rules as normal. No weird foldy-outy stuff. But, yep, 4x6 is fine. And if you find ways to, make information where it's not just a bunch of writing, that could be helpful too. It's hard to put draw little pictures on there, but on the other hand, it does give you information. So you can figure out too where you feel like you're weak and then prioritize that for your card, right? Instead of wasting time writing things on there about, vertebrates, which maybe you know about already. Jump off. It's not going to take you 2 hours and 50 minutes unless you're just, really slow. Don't worry. You're not going to take that long.